Hey guys, welcome back to another tutorial video. In this video, we are going to discuss about Helicobacter pylori, which is a gram-negative bacilli which causes the peptic or stomach ulcer, which is the leading disease and the cases are reporting nowadays. Now we shall talk about the general characteristics about Helicobacter pylori. It was first discovered by Barry and Robin in the year 1980. It was previously called as Campylobacter pylori. Under the Gram's classification, it's a gram-negative spiral-shaped bacilli. Capsule and endospores are absent. In the case of respiration, it requires little amount of oxygen to grow. Optimum temperature 37 degrees Celsius. Optimum pH 6.9 to 8. As per the report, the natural habitat of Helicobacter pylori, human stomach alone. The mode of transmission of Helicobacter pylori is transmitted via person to person by saliva or the fecal contamination of food and water materials. The factors which is responsible for the virulence of Helicobacter pylori is flagella for movement, urease, an enzyme which neutralizes the gastric acids, adhesion molecule which supports for the attachment, lipopolysaccharides which induce the inflammatory response. The last one is the cytotoxin associated gene which attack the neutrophil and contribute to gastric ulcers. Now we shall discuss about the pathogenesis of Helicobacter pylori. In the first step, we could see the first layer is the gastric juice, followed by the second is mucous layer, and then third the epithelial cell lining of stomach. So with the help of the flagella, the Helicobacter pylori that reaches towards the epithelial cell lining. In the second step, with the help of the adhesion molecule, the bacteria that attaches to gastric epithelial cells of stomach, and then it produces the enzyme called urease, which converts the neighboring urea into ammonia that neutralizes the gastric acid. In the third step, the production of ammonia that leads to form a barrier around the bacteria. In the fourth step, the bacteria that invade into the epithelial cell lining of stomach and produces cytotoxin associated gene, which attacks the neutrophil and a release of protease which allows the gastric acid to enter into the stomach cells which contribute the gastric ulcers. This is how the H. pylori causes peptic or stomach ulcer. The clinical symptoms of stomach or peptic or duodenal ulcers that includes abdominal pain, bloating and flatulence, fatigue, nausea, vomiting, diarrhea, bloody stool, heartburn, bad breath and full feeling after consuming little amount of food. These are all the symptoms of stomach or peptic or duodenal ulcers. Now, how to identify the Helicobacter pylori from the given clinical samples. In the microscopic examination under the gram staining, it shows pink color, spiral shaped gram-negative rods. It is motile in nature. In the case of histological examination, tissue biopsy prepared by using giemsa stain or silver stain and examined under the microscopic examination. The serology test that includes ELISA to detect the antibody against the H. pylori, 16S rRNA sequencing, and the final one is polymerase chain reaction. These are all the various laboratory diagnoses of H. pylori. In the biochemical test, it shows positive for catalase, oxidase, and strong positive for urease test. In case of IMVIC, it shows negative results. That includes indole, methyl red, Vogus proskyde, and cytoutilization test. For treatment, the combined therapy with various antibiotics that includes macrolides, beta-lactam, and omeprazole for 2 weeks. Human vaccines are not currently available. The prevention is practicing the good hygiene practices. So far we have discussed about the H. pylori general characteristics, the mode of transmission, how it causes the peptic ulcer, and then treatment. Thank you.